This is Screen Capture Video Unit 11.1, and this follows Chapter 9 out of Biology by Campbell et al., published by Pearson. This unit has to do with heredity, and a good place to start with heredity is talking about dogs. A lot of people have dogs. They're one of America's favorite animal, and there are many different breeds of dogs to suit different tastes and needs. In the past, dogs were bred according to filling some kind of a need for family or hunting or protection. In more recent history, say in the last 100 years, dogs have been bred partly for aesthetics and somewhat entirely for aesthetics, just to look at and have as a pet, not necessarily as a service animal. But when you look at dogs, the different breeds, they all make up one species. They're able, supposedly, to interbreed. Technically, that would be difficult, and researchers tell us that it's difficult for a small dog like a Chihuahua to be bred by a large dog such as a Dalmatian or Great Dane. One of the problems would be that the smaller breed would have trouble carrying the pups to full term. And so, even though they're considered one species, there's a tremendous amount of difference amongst the breeds. Some are well over 100 pounds. I think the world record for some of the Mastiffs may be over 200 pounds, I don't recall. But very large dogs. And then you have small lap dogs such as the Chihuahua and the Poodles. But one of the questions that people would ask is, how do those traits come to be? And how is it that out of one species there are so many different breeds of dogs? They look so different from each other. Well, that all has to do with heredity, the passing of information, the genetic material, and how it's expressed from one generation to the next. As we go back historically, we take a look. The Greeks considered reproduction and heredity. Socrates had a little bit to write about it. Hippocrates had a little bit to say about it. And Hippocrates believed that there were particles gathered from different parts of the body called pangenes, which would travel to the sex organs to be collected in what today we would call sperm and ova, though at this time the Greeks would not have understood the idea of sperm and ova as we understand it today. But these different pangenes would be collected from the body and then assimilate to form a new person. And whatever characteristics the father had and characteristics the mother had, they would just get put into the offspring. And so one of the problems that was seen with this was if the father had lost his arm in battle or in a farming accident, it would not follow that the child would be born without an arm. That would just follow superstition, but not science. And so there were some disagreements. Socrates had some disagreements with this. And eventually, around the 1800s, there was a pangenesis theory. There was a blending theory. The pangenes were part of what came to be known as the pangenesis theory. The blending theory in the 1800s stated that physical traits from the mother and father were blended into the offspring. And that was how they described seeing part mom, part dad in the offspring. And that seemed to go with some people, except it didn't explain how some traits might show up after 3, 4, or more generations in the offspring. How did that happen if it was blending? They should have seen that in previous generations where it seemed to have disappeared. Mendel came along in the mid-1800s and he eventually published in 1866 where he disproved those two theories. And Mendel studied flowers. Gregor Mendel was a Roman Catholic clergyman in the Austro-Hungarian Empire, part of it. And he specifically worked with pea plants, and he manipulated the pollination process, the breeding of those pea plants. To his credit, he took some difficult classes at the university, including math, quite a few math classes, chemistry, some physics, as well as the other science classes that we call biology today. And he used those, especially the math, in calculating what he was seeing in the breeding experiments that he was doing. And he wrote and kept good records. And that's one of the reasons why he's recognized as the first geneticist. You could go back in time and you could read about other scientific works, but if they didn't do a good job recording them, then they weren't given much notoriety. He specifically understood the flower parts. When you consider a flower of a pea, and this is true of many flowers, there's the carpel, which some people liken to the female sex structure of the plant, and the stamens being the male parts, where these are producing pollen, which some would liken to spermatozoa. And the pollens can land on the carpel, and the flower can fertilize itself, self-fertilize. It's possible. And then the pollen grain goes down and unites with the ovum, and peas can eventually be produced. Well, Gregor Mendel recognized this pollination process, and so he would take pollen from one plant, and he'd use a brush, gather the pollen, and then he'd brush the carpel of a different plant and pollinate it. And what it's not showing here perhaps is, it does say he removed the stamens. He'd remove the stamens from the plant that he was pollinating so that the plant couldn't self-pollinate. And any pollination that was occurring was coming from the plant he gathered pollen from. And so he could manipulate the breeding and then watch what happened in the offspring, in the succeeding generations. Now it helps if I introduce some terms here. So I'm going to scroll down, and I'm going to come back up and take a look at some of his experiments that he did. With the terms, in the past, in the last unit, Unit 10, I talked about autosomes and sex chromosomes. The autosomes are the 22 pairs of chromosomes, 1 through 22. That would be 44 chromosomes in total because of the 22 pairs. And then there are the sex chromosomes, X's and Y's. The female carries 2 X's, usually, and the male carries an X and a Y, usually. Genetics is the study of heredity, the science of heredity. And as I mentioned, people have been interested for millennia of what traits would be passed on to the offspring. For many millennia, fathers especially wanted male children. But even beyond having male children, they wanted to see certain characteristics in their children. And those characteristics, those traits have been written about historically, and we have an idea of what people look like as they were described. But some of the phenotypes aren't so easily seen or noticed. Character. Some authors will use the term character, and this is sometimes referred to as a heritable feature, such as flower color, eye color, whether the earlobes are attached or whether they're free, a widow's peak or not having widow's peak, the hairline, so to speak, the arch structure of the feet, those hair on the fingers, or not having hair. Those are characters. The traits would be more specific. That would be, if you're talking about flower color, whether the flower color is purple, or whether it's white, or pink, or yellow, or green, whatever it might be. You'll hear me use the term or expression true breeding. And true breeding means where, especially in peas, where they self-fertilize and they produce offspring that are identical to the parents. So no matter how many times the flowers self-pollinate, the offspring always look like the parents. So that tells you that there's just one allele for a given character, as I've described it so far. A hybrid is when you take 2 different parents having alleles or genes for 2 different traits, for a given character. You breed them, and the offspring are called a hybrid. Now, that doesn't necessarily mean that both traits are going to show up in the hybrid. It may be that only one of the traits shows up, but that's a hybrid, and that's sometimes referred to as the F1 generation, which I'll talk about in just a little bit. You'll see the letter P, capital P, and usually there's not a 1 next to it, but just a P by itself, and that stands for parent or parental generation, and so that's where you start the breeding process generally in an experiment. You'll start with the parents because you know what they are. The F1s, that would be the first filial generation, or the first offspring from the parent, or whatever the parent produces would be the F1 generation. And then if you take the F1 individuals and you breed them with each other, you get the F2, and that's the second filial generation. Test cross. This is done when you have an unknown and you're not sure what the genetic makeup of the unknown is. You cross it or breed it with a homozygous recessive. And I'm going to get into homozygous and recessive here in a little bit. But by crossing with the homozygous recessive and looking at the offspring, then you can go back and determine what the unknown is, what the genotype is for that. So the test cross is really handy to give you an idea of the genetic makeup of an unknown. Homozygous. When you think of homo, you think of the saying, this is where the two alleles for a given trait are the same. So it would be purple for one parent, purple for the other, or white and white. Heterozygous, other, that means where the individual would have, say, a gene for purple and a gene for white. Or if you're talking about the parents, one would be white and one would be purple. Dominant, this refers to the allele that shows up or that is expressed when the other allele is recessive. So for purple and white, purple would be dominant, and so if an individual plant is carrying purple and white alleles or genes, the purple shows up and the white doesn't. The white would be described as being recessive. It's masked or dominated by the purple. So if there's a dominant gene or allele, the recessive wouldn't be seen. When I talk about alleles, that's an alternate version of the gene. So there's a gene for purple, there's a gene for white, or we could say an allele for purple, allele for white. And the trait is actually what we see of those. The law of segregation, independent assortment, I think I show that in an illustration just a little bit, or in the next video. And this is where the alleles for a given trait are able to separate in meiosis so that gametes can be produced, and one allele can go into one gamete, another allele can go into another gamete, and they're not linked. Eventually I'll talk about linked genes, and this is where the gametes, or the alleles do not separate. But Mendel came up with this law of segregation or independent assortment, and he was correct in seeing that in unlinked genes. I'm going to end this video here, and when I come back I'll talk a little bit about the illustrations that show some of Mendel's work with pea plants and some other crosses.